Okay, so this video we're going to talk about photosynthesis, and we're going to have a look at an exam question. So we're going to start with the light independent reactions of photosynthesis. Now this really is the Calvin cycle. We know the Calvin cycle occurs in the stroma of a chloroplast. Now the Calvin cycle is a set of reactions that ultimately is going to allow glucose to be formed, but it does require two products of the light dependent reactions. So the light dependent reactions, or the LDR reactions, occur at the thylakoid membrane. Now we know ATP and NADPH are two products of the LDR that are going to be needed for the light independent reactions of photosynthesis. So before we actually get to the exam question, it's probably good practice to remind ourselves of the Calvin cycle. So if you guys would like to pause the video and have a quick go at the Calvin cycle, and then just click play, and you can compare it to the one that we've got on the screen. So the Calvin cycle then occurs in the stroma of the chloroplast. Now we know that carbon dioxide is the inorganic molecule that's required. We know carbon dioxide can simply diffuse into the palisade mesophyll cells, and it can then diffuse simply across the two membranes of the chloroplast into the stroma. So it's going to simply diffuse from higher to lower concentration into the stroma of the chloroplast. Now the carbon dioxide reacts with a 5-carbon organic molecule called RUBP. Now that's called ribulose bisphosphate, but for AQA you can just say RUBP, but you must know it's a 5-carbon. Now the 1 carbon of the CO2 reacts with the 5-carbon of the RUBP, and that is catalyzed by an enzyme called Rubisco in the stroma of the chloroplast. Now Rubisco, as long as you know the name of that, is okay. It's an enzyme. It catalyzes carbon dioxide fixation, but it does stand for RUBP, or ribulose bisphosphate carboxylase oxygenase. So the carboxylase reaction is the fixation of carbon dioxide onto RUBP. Now that does actually form a 6-carbon compound, but that's very unstable, and that splits into 2 molecules of glycerate 3-phosphate. So the end product of CO fixation, step number 1, is 2 molecules of glycerate 3-phosphate, and you can shorten that to GP. So for every 1 molecule of RUBP, you will have 2 molecules of GP, so double the concentration. Now we know GP, glycerate 3-phosphate, is reduced to triose phosphate. Now we can't say GP is converted to triose phosphate because we have to use the term reduced, and we have to know how it's reduced. So one of the molecules from the light dependent reactions is ATP. The hydrolysis of ATP produces ADP and inorganic phosphate. Now the small packet of energy that gets released is enough to drive the reduction reaction of GP to triose phosphate using the hydrogen that is released from NADPH. So that hydrogen is used to reduce GP to triose phosphate with the small packet of energy from ATP hydrolysis. Now this will reoxidize NADP, and that will then go back to the LDR reactions at the end of the electron transport chain, and that will then become reduced once again. Now we know triose phosphate is a 3-carbon compound, organic compound. 2 of these triose phosphates are required to synthesize 1 glucose. Now we know glucose is a hexose sugar, and that means it's got 6 carbons. So glucose is C6H12O6. Also, the triose phosphate in the stroma of the chloroplast, it can also be used to regenerate the RUBP, the 5-carbon. Now this is going to require some more ATP, but hopefully you can see that the hydrolysis of ATP will release ADP, but the phosphate and the small packet of energy is used to drive the regeneration of RUBP. So in the LIR reactions in the stroma, there's 3 processes that happen: carbon dioxide fixation, the reduction of GP to triose phosphate using ATP and NADPH, which are both produced from the LDR reactions in the light at the thylakoid membrane, and the regeneration of RUBP, the 5-carbon compound, from a number of different triose phosphate molecules. And that again requires some ATP. Now, the thing that you guys should hopefully take away from this is that the triose phosphate, yes, we said 2 of those are required to synthesize 1 glucose. The glucose, it could be alpha or beta glucose, can then be used to synthesize either starch, so you can have starch grains in the chloroplast, or beta glucose could be used to synthesize cellulose, which you guys have to be able to describe. So straight polysaccharide chains cross-linked by hydrogen bonds to form microfibrils. But the triose phosphate can also be used to build disaccharides via the glucose. So glucose and fructose make sucrose. Or the triose phosphate, when it's converted to glucose, that glucose can be used to build nucleotides. And it can even be used to build amino acids in the plant cells. Some of the glucose can also be used to generate chlorophyll. So most of the organic molecules that you get in plant cells are synthesized within the plant cell itself, and they're going to use the glucose as a starting molecule for that. But please be able to talk about the 3 processes in the LIR: CO2 fixation, GP being reduced to triose phosphate, and triose phosphate being used to regenerate RUBP. Now, a side point to this then, if you have 6 cycles of the Calvin cycle, that means 6 molecules of carbon dioxide will be fixed. So we know the balanced chemical equation, we've got 6 molecules of CO2 that will be fixed, and that will allow the single production of 1 glucose. So from 6 cycles, we'd actually end up with 12 triose phosphate. Now, triose phosphates, 2 of them are used to build glucose, which will leave 10 triose phosphates. Now, because triose phosphates are 3 carbons, if there's 10 of them, that's collectively 30 carbon atoms. Now, with 30 carbon atoms, 6 5-carbon compounds can be made. So 6 5-carbon compounds is also 30 atoms. So there's a series of chemical reactions catalyzed by enzymes that convert the 10 triose phosphates with their 30 carbon atoms into 6 RUBP with their 5 atoms each. Now, if there's 6 RUBP and there's 6 molecules of CO2, we can have 6 carbon dioxide fixation reactions taking place, which we know are catalyzed by Rubisco. Now, the next slide, then we've got a typical exam question. So we are going to get some data on a graph or maybe a table, and we're going to get quite a lot of information that we have to annotate before we even attempt the answer. So the key here is to pick off information from what we're actually given in the question. So if we look at the graph to start with, obviously you're going to look at the 2 labels first. You need to look at the y-axis on the left. So what they've measured is the mean concentration of triose phosphate, and that's in the stroma from the light-independent reactions. They also have on the x-axis the time after the light is switched on. So it goes up in 4-hour increments. So what they've done is to measure the mean concentration of triose phosphates from the time the light was switched on, time point 0, then after 4 hours, then 8, then 12, then 16 hours. And we can see there's 2 groups here. There's a control group, and there's also an experimental group. So let's go back into the information at the top and see what they tell us. So it says they grew sugar beet plants under the same conditions with their roots in liquid growth media containing all the necessary nutrients. That means they used a bunch of plants with a large sample size, ideally. The roots were put in growth media with nitrates, phosphates, magnesium, iron, everything the plant needs to be able to grow. 10 days before the start of the experiment, they transferred half of these plants to a liquid growth medium that was exactly the same, apart from it had no iron. The other half of the plants were left in the same liquid culture media. So the independent variable for this experiment is the lack of iron and their growth for 10 days versus the presence of iron in the growth media for those 10 days. The scientists then measured the concentration of triphosphate produced in the stroma of the chloroplasts, in the experimental plants with no iron and in the control plants that had access to iron that they would have absorbed by active transport. So we could make an annotation on the question, which is what you should do. So what they're testing, the independent variable, is the lack of iron versus the presence of iron. And what they measure is the mean concentration of triphosphate from the stroma of the chloroplasts. So they're going to measure the mean concentration of triphosphate from the stroma of the chloroplasts in the control group that had access to iron and compare that to the experimental group, which did not have iron in the liquid growth media. Now then it tells us that actually after these 10 days, they put both groups of plants in the dark for 6 hours. Now we could annotate this because we know in the dark, there's no LDR reactions. So that means that there's a lack of ATP and there's also a lack of NADPH, because if there's no light, there's no photoionization and there's no photophosphorylation at the thylakoid membrane. So after 6 hours in the dark for the control group of plants and the experimental group of plants with no iron, they then switched the light on for 16 hours and they measured the mean concentration of triphosphate. Now we know as soon as that light is switched on, the chlorophyll in the thylakoid membrane can absorb red and blue light, which allows the LDR reactions to proceed. Therefore, ATP and NADPH may both be produced. So when they switch the light on after the 6 hours in the dark, that is time point 0. So time point 0 on the x-axis, is when they switch the light on and they leave it on for 16 hours. Now, what you guys should be getting in the habit of doing is circling the data and using the data off the graph or a table if a table is given. So here we can see that actually at time point 0, when they switch the light on after the 6 hours in the dark, the mean concentration of triphosphate is very similar for the control plants with iron compared to the experimental plants with an iron deficiency. So it's not 0. The mean concentration of triphosphate is not 0. So the ATP and the NADPH have not been completely depleted by the 6 hours in the dark, but it's actually allowed quite a low level of triphosphate to be produced. And it's very similar, the key point is very similar for both groups of plants to start with at time point 0. So if you guys want to quick go at the questions then, so 5A to 5B, feel free to obviously pause the video if you want to quick go at the questions yourself. And then once you've finished, if you just click play, and then I'll go through the answers with you. Okay, so if we start with question 5A, number 1, it says the experiments were carried out at a high carbon dioxide concentration, and then it says explain why. Well, you can only have one independent variable for any experiment, and that's what you set as a scientist. So here they're comparing the presence of iron in the liquid media, which is our control group, to the absence of iron in our experimental group. That's the independent variable. So what we don't want is to have a low carbon dioxide concentration, because then that could interfere possibly with the results. Now, we wouldn't know if we had a low carbon dioxide concentration if the changes in triphosphate concentrations were due to the low carbon dioxide or the lack of iron. It would be a confounding variable. So as long as we've got a very high concentration of carbon dioxide, we know that it's not going to be limiting in terms of the Kelvin cycle. So it allows us then to investigate the effect of the lack of iron. So if we look at the answers for question 5A, number 1, if carbon dioxide concentration is nice and high, that is not going to become a limiting factor for the light independent reactions. So we are going to assume that that is not going to impact the production of triose because we're not investigating the concentration of carbon dioxide. Right, if we look at question 5A number 2, it says, explain why it was important to grow the plants under the same conditions up to 10 days before the experiment. So if we go back to the text at the top, okay, it says 10 days before the experiments, they transferred half the plants to a liquid growth medium containing no iron. Okay. So this was the 10-day period with no iron compared to with iron. Prior to that, though, they actually grew the plants in exactly the same liquid culture media prior to the 10 days. Now that's going to be really important because when you start the growth in the 10-day period without iron compared to with iron, we know that allows us to see any difference in the mean concentration of triose phosphate in the light is due to the absence of the iron. So that is our independent variable that we're investigating. So the two different sets of plants, if you like, were grown in the same liquid culture, but then 10 days prior to the start of the experiment, okay, we switched half of them to the liquid culture with no iron. So this is going to be our independent variable. It allows us to look at the absence of iron on the mean production of triose phosphate. Okay, the next question then is, the plants were left in the dark for 6 hours before the start of the experiment, and then it says explain why using our knowledge of biology. So the plants were left in the dark for 6 hours. Well, in the dark, no LDR reactions can take place. Now, if the LDR reactions cannot take place, it means there's going to be a lack of production of ATP and also a lack of production of NADPH because they both require the light to be produced. So we know photoionization, photophosphorylation, and also photolysis occur in the light. But if we look at the graph, we're going to use the data there. So we can see after 6 hours in the dark, there's actually a low concentration of triose phosphate, mean concentration of triose phosphate, both the control group and the experimental group. So it's about 7 or 8 arbitrary units. So the mean concentration of triose phosphates will be low, but very similar to the control group and the experimental group at time point 0. So time point 0 is where the light is switched on. If you look at the mark scheme on the right, it does say reject, so all triose phosphate is used up. So we can see after 6 hours in the dark, there is still a small concentration of triose phosphate present in the stroma for both the control group and the experimental group. So they rejected the idea that there was no triose phosphate, because we can see from the data in the graph that there is a little bit. Okay, it then says iron deficiency reduces electron transport. Use this information and your knowledge of photosynthesis to explain the decrease in production of triose phosphate in the iron deficient plants. So we've now got a link, okay, a lack of the electron transport chain, or a lack of electron transport in the thylakoid membrane, okay, with the decrease in production of triose phosphate in the iron deficient plants. So if there's iron deficiency, there's less photoionization and less electrons moving down the electron transport chain. We know that normally the electrons lose energy as they move down the electron transport chain, and this activates a proton pump. Now that proton pump is responsible for generating an electrochemical gradient between the stroma and the thylakoid space. Now normally, once that electrochemical gradient has been generated, the protons can only diffuse back into the stroma through the stalk particle, which contains ATP synthase. Now normally, NADP would pull the electrons through the electron transport chain and become reduced, and the electrochemical gradient will be used in photophosphorylation to produce the ATP at ATP synthase. But if there's a reduction in the electrons moving through the ETC, which happens with the lack of iron, then there's going to be a lack of photophosphorylation, because of the diminished electrochemical gradient. And there's also going to be a lack of NADPH. Now, if there's less ATP and less NADPH, it means the reduction of GP to triose will not occur to the same extent. So that GP reduction step in the Calvin cycle is not going to proceed as much as it would in the control group. So we're going to link the LDR with the LIR. So if there's less electrons being able to move through the thylakoid membrane, through the electron carriers, the electron transport chain, there's less ATP produced by photophosphorylation. There's less NADPH being produced at the end of the electron transport chain in the thylakoid membrane. So we should really use the words less in both answers. ATP and reduced NADP are normally produced in the light-dependent reactions, like we just mentioned. But because these cannot proceed, there's lower amounts of ATP and NADPH, which has a knock-on effect for the LIR reactions, the Calvin cycle. So there's less GP being reduced to triose phosphate that requires the hydrolysis of ATP and the hydrogen from NADPH. So we can see 3 of the 4 answers there should really have the word less within them. So this is an application question. Now we can actually see from the dotted line, the dotted data on the graph, the iron deficient plants from time point 0 at all time points over the 16 hours in the light, the iron deficient plants have a lower mean concentration of triose phosphate compared to the same time points for the control plants that had iron in the light. So we know iron is a very important mineral ion that allows the electron transport chain to work in the thylakoid membrane. Proteins can function correctly and their redox reactions can occur as normal. So less GP reduced to triose phosphates for mark point number 4. Now the last question, 5C, said iron deficiency results in a decrease in the uptake of carbon dioxide. Explain why. Now we know iron deficiency up to this point means there's less ATP and there's less NADPH. If there's less reduction of GP to triose phosphate, there's a lower mean concentration of triose phosphate. So with the lower mean concentrations of triose phosphate in the light, it means there's less RUBP being regenerated, not only because there's lower triose phosphate, but also because there's less ATP because of the lack of the electron transport chain functioning correctly. So if there's a lack of triose phosphate and a lack of ATP, there's a lack of regeneration of RUBP in the stroma. Now, if there's less RUBP present, it means less carbon dioxide can be fixed to make glycerate 3-phosphate or GP. So less carbon dioxide is being used up in the fixation step because of the low concentrations of RUBP. Okay, so the mark scheme answer is less triose phosphate is used to regenerate RUBP, and therefore because there's a lower concentration of RUBP, less CO2 will combine with it. There's less CO2 fixation with RUBP, and therefore there's a smaller uptake of carbon dioxide. Okay, and that's the end of this video. If you feel that was quite helpful, please just give us a like and subscribe as well. So I hope that was useful.